Welcome to Practical Skills Planning an Experiment. This is our first set of practical skill notes, and it's a really important one because we need to understand how science works, really to understand everything we're going to talk about this year. And of course, you need to understand experimental design for the ACE exam as well. And here are your two favorite marine science teachers ready to do labs with you. And let's go ahead and get started understanding experimental design. As always, it's really a good idea to understand what we're about to learn. And here are the main ideas. Experimental design includes the formation of a testable hypothesis, the identification of variables, and the design of an experiment to collect the appropriate data that is reliable. Scientists have a responsibility to act ethically and safely during any investigation. And of course, there is a series of specific targeted learning goals that go through each step that we're going to walk through during the notes. Let's start with the word science. What is science? The word itself is derived from the Latin word scientia that translates to knowledge. Science essentially is a systematic and logical approach to discovering how things in the universe work through testing and analysis. Now, there are a number of characteristics of science, and we're going to go through some of them here. First, science asks questions about the natural world. We have to deal with what's right in front of us, what we can collect data from. It aims to provide accurate explanations and reliable predictions, meaning that when we come up with a hypothesis or a theory, for example, which we'll get into a little later, we want to see, do they predict correctly the outcome? They can only work with testable ideas. So things that we can't actually get data on, we can't actually test, really doesn't fall on the line of science. It relies on evidence, not dogma, popular opinion, or tradition. A lot of things that we just thought or assumed occurred a certain way, we have found do not do so when we actually collect the actual data. And that sometimes makes people uncomfortable because they're so used to thinking of it in a traditional way. And this is where sometimes we have a bit of a disconnect sometimes. Science relies on scientific community for building consensus. This is really important. What is the scientific community? All the scientists around the world that put out their findings in published papers and share their results. And everybody gets to read it, dissect it, and try and determine whether or not a particular study was done correctly, if maybe somebody missed something, and then allows other scientists to follow in the footsteps of others and retest these ideas to see if they can disprove them. This process helps to eliminate bad science, but it is also a very lengthy process. Science takes some time, and this is also something that people don't always understand. And of course, scientific ideas lead to ongoing research. We learn about something, then we ask new questions we didn't even know to ask before, and that continues the process on and on. Now let's focus on this whole idea of practical skills. Now, scientists use a set of skills to investigate natural phenomena, and that's exactly what you're going to do. You're going to be applying various skills that we learn to lab and field investigations throughout the year. You can see some images of scientists doing various different types of lab and field work. Now, the things you will be focusing on by the end of the year, you should be able to plan experiments, present your observations and data clearly, evaluate the method and quality of your data, and draw appropriate scientific conclusions. Now, the first step in the scientific method will be to define the problem. This usually starts with an observation. Someone sees something they don't quite understand, and they ask a question. That's essentially what scientists start out with. Now, on the picture to the left, you have an image of something you might not be very familiar with. If you were in scuba diving on a reef and you saw this in front of you, you might ask some questions. You could ask, what is it? Is it an animal? Is it a plant? Is it alive? How does it eat? You could ask all of these questions. And you then could design an experiment to further your information and knowledge about that to answer some of those questions. That is essentially the process. Just so you know, that is an animal. That's flowery looking yellow thing. It is called a crinoid, and it's actually related to sea stars. Now on the right, there's another image of a fish called a rock beauty and 2 shrimp called scarlet ladies. Now, you might ask, if you were looking at this scene, well, what's going on? How is the fish interacting with the shrimp, and how are the shrimp interacting with the fish? What is actually happening? Now, in this particular question can be answered by watching what the shrimp are actually doing. They're like picking on the fish and bringing stuff to their mouths. They're actually picking off parasites. This is a symbiotic relationship going on. The fish is essentially getting cleaned up. Now, my point with this is just to show you that this is the starting point of scientific investigation. You see something, you don't quite understand it, you try and figure it out, and you ask questions. Now you start to move into what a hypothesis is. A hypothesis is formed by trying to explain the observation. You want to answer the why, and that's essentially what scientists spend their entire lives doing. So what is a hypothesis? It's an explanation of an observation that can be tested through experimentation. This generally leads to what we call a prediction, a statement of the expected results in an experiment based on the hypothesis being tested. Now, there are lots of misconceptions when it comes to a hypothesis. Most people relate this term with a guess. The reality is a hypothesis is based on experience, past scientific knowledge, and it has explanatory power. It is not something we just throw around lightly. Now, just so you're aware, on the ACE exam, you may be given an observation and will be expected to formulate the hypothesis and prediction using your knowledge of marine science. You may also be asked to provide your prediction in the form of a graph. And of course, we will practice these things throughout the year. Step 2 is all about planning the experiment. You're going to design an experiment in order to test the hypothesis that was created. Now, this should produce what we call quantitative data, and we'll get into that in a little bit, that will be used to support or refute your hypothesis. Essentially, that's what we do. We either support the hypothesis or we refute it. We never actually say the hypothesis is 100% accurate or correct. Now we need to take a step back here and look at variables, because you're going to have to determine the variables in your experiment. Now, you've probably heard these before, right? The independent variable is the variable being changed in an experiment. It's the manipulated variable. The dependent variable is the variable being measured in an experiment. It's the responding variable. All right, so you change something in the independent variable and you see what happens to the dependent variable. Now, we also have what we call control and confounding variables. Now, control and confounding variables are essentially the same thing. They're variables that are not being tested, but must be kept the same in case they affect the experiment. Now, in this image, you'll see us doing a little lab with water and alcohol to see which of the two substances travels up a strip of paper towel the farthest distance. Now, we're testing the polarity of water in this particular experiment. Now, obviously, the thing we're changing, the independent variable, would be exposing the paper towel to either water or alcohol, right? Now, the dependent variable would be what we're measuring, which is the distance that substance travels up the paper towel. Now, in terms of a control variable, we're looking for things like the same length of paper towel, the same volume of water and alcohol in either beaker, the same size beaker. Essentially, anything that could change the experiment is your control variables. You want to keep them the same. Now that we've defined our variables, let's backtrack just a little bit and talk about writing a hypothesis. So you can use the standard if, then, because format if you would like. It helps you kind of identify those variables and write it in a succinct way. If you're going to do this, this is how it works. If the independent variable, whatever that is, you then describe how you're going to change it, then the dependent variable, and then you describe the effect, and then because, and you provide the explanation. So let's walk through this in an actual hypothesis statement. If the surface area is increased, then salt will dissolve faster because it will have more contact with the solvent per unit of surface area. So in this case, if the surface area is increased, that is the independent variable, and that is how we are changing it. We're increasing the surface area. Then the salt will dissolve faster. So the salt dissolving is the dependent variable. How fast is it going to dissolve? And the explanation has to do with more contact with the solvent, which happens to be water, which is the universal solvent. Now that you understand your variables, you've got to set up your experimental and control groups. Now, the experimental group is the group within an investigation in which the independent variable is being changed or manipulated. So that's where you would be, for example, increasing the surface area or using water versus alcohol in the experiments previously described. Now, the control experiment is a group within an investigation or study that receives exactly the same treatment as the experimental group, with the exception of the variable being tested or changed. Now, an example of this is the first lab I generally do each year, where we put calcium chloride in water and measure the temperature increase, because as calcium chloride dissolves, it's exothermic, it releases heat energy, right? So the control is essentially the same exact setup as the experiment, except I don't put calcium chloride in there. And I don't do that so that I can measure the temperature change. Maybe the room got warmer over the experiment, and that's the temperature change I saw with calcium chloride, and it really didn't do anything. I wouldn't know that unless I had the control group, which was just water, and measuring that throughout the entire time as well. So control groups are incredibly important to the validity of scientific experiments. Another thing you're going to have to do is choose the appropriate techniques. The appropriate techniques to measure the various different variables accurately. Now, I've given you an image here of all sorts of scientific laboratory tools, but there's also a whole nother set of tools for the field. What are you going to use to measure them, and making sure that measurement is as accurate as possible? Now, in terms of choosing the appropriate techniques, there's a series of things that we can talk about, many more things than we can fit in this slideshow, but I'm going to give you some examples from a laboratory experiment-based setup. Now, in the lab, there are more options to control and manipulate variables just because of the nature of being in a controlled environment. Now, here's some examples. A water bath, for example, a container of water heated to a given temperature, which can be used to either vary or control temperature. Now, when we do the productivity experiment, where we do rate of oxygen bubbles being produced from a little sprig of a water plant called Alodia, I use a larger beaker with a larger volume of water as a water bath to help stabilize that temperature. A buffer is a type of solution which can maintain a relatively stable pH, and that's important. You don't want your pH swinging all over the place if you are testing something at a specific pH. Now, we can also control carbon dioxide in water, for example, and oxygen in water, often by making sure they're either available in excess and not limited. You can do this by adding hydrogen carbonates to the solution or aerating with bubbles in an air pump. Controlling light intensity. You can move a light closer or farther away from the subject that you're testing. You can control the wavelengths, things like that. You can use the correct equipment, of course. For example, if you are measuring something that's a very small volume, you want to use the smallest volumetric beaker or even a graduated cylinder or something to measure it more accurately than something very large of small volume. So you have to pick the right equipment for the right application. When we work in the field, it's a little bit more challenging because the environment has variability that we really can't do a whole lot about, such as temperature fluctuations, light, things like that. Measurements need to be taken of all possible variables, and this essentially hopes to identify maybe any anomalies later on in the data. You also want to make sure you are applying random sampling where appropriate to avoid bias in the data. Now, as we talk about appropriate techniques, we need ways to measure the variables, and here's a table that gives you some of those. Temperature, for example, you can monitor through a thermometer. The most accurate are the digital thermometers for pH, using a pH indicator, a pH probe to measure the pH. Light intensity, just proximity can do that, but you can also use various light sensors. Volume, measuring cylinder, a pipette, or syringe to measure volume of a liquid. Mass, you can use a balance. They're digital balances, of course. For time, you can use a stopwatch. Most phones have stopwatches, so that makes it easy for you guys. And of course, distances. For shorter distances, maybe a ruler with millimeters. For longer distances, maybe you would use a tape measure in meters, things like that. Anytime you are conducting a scientific investigation, you want to make sure you talk about safety and ethics within your work. Now, for a safety standpoint, you need to evaluate the hazards and risks associated with your experiment. Hazards are something which could cause harm or damage to your health. Risk is a chance of this damage happening and how severe that harm might be. Now, this includes how to safely use equipment, proper disposal of chemicals, things like that. You can imagine, for example, if you're scuba diving for a particular project, you need to make sure you're certified and all the safety protocols are being followed. If you're working with an animal, you might need to protect yourself from the animal itself. You may need to protect the animal itself from harm while you're conducting the experiment. These are all things you have to consider. You may also need to actually obtain permission to do whatever work you're doing. For example, field work may require permission of the local authorities. If you're working with animals in the field, you may need permission to do that too. So obtaining permits for these types of activities is probably important. You might want to consider the weather or the conditions out there in the field as well. And of course, chemicals have specific procedures that are guided by state and federal regulations that you must follow. Here's an example of a table giving you some hazards and risks. For example, corrosive chemicals enter the eye or harms the skin. So you want to wear gloves to protect and protect your eyes and that kind of a thing. Glass equipment, it can break and enter the eye or cause cuts in the skin. So you want to wear goggles and make sure to keep glass equipment away from the edge of the bench to avoid breakage, things like that. Hot equipment burns the skin, right? So you want to take care with hot equipment, use tongs to hold the glassware, avoid touching hot bulbs, things like that. Now, when we're dealing with live animals, we have to deal with ethics that relates to how you treat these other living organisms and avoid causing them harm. You don't want to cause harm. You don't want to release exotic species in areas where they're not supposed to be, and you don't want to release toxic chemicals or pollutants that might affect the area as well. Now, we deal with a variety of animals, everything from invertebrates like these sea urchins on the left, or even the great white shark on the right, where they are tagging them for release again so they can track it. Now, when you're tagging an animal that large, you have to make sure that those animals are going to survive. And they're very efficient at doing it very quick. They take blood samples, they put the tag on, and they get them back out in the water. And they have a very high survivability rate. So the cost in terms of the stress to the animal, it outweighs the benefit of what we gain out of that data that helps us protect their species. Now, getting back into the experimental design here, you want to determine the number of measurements you need to make. First, you need to choose the range of values or levels of your independent variable. If possible, your range of values or levels should be evenly spread as well. For example, here we're looking at the effect of light on productivity in that beaker, which has a little piece of plant in it, right? So we can divvy it up. The independent variable would be how far the plant is away from the light. And we can specifically set distances that we're going to look at. Those are your different levels. Now, in the intertidal zone over on the right, we have various different, a transect line, and we can do distances from the water's edge away from the water and look at those distances as levels of your independent variable. You also need to determine the number of replicates at each level. How many times are you going to do each experiment at each level? There should be a minimum of 5 to 10 replicates in order to compare results effectively. If you do less than 5, you're really not going to be able to make any valid conclusions. So replicates are really important, and they identify anomalous results, which is also a really important thing. Now you've gotten your experiment designed all figured out and you're ready to collect data. Now let's talk about the data, specifically uncertainty, because all data has a certain amount of uncertainty. You want to make sure your results are as reliable as possible and that they are replicated by others. That's why you take such care in designing your experiment. So there is something called an anomaly or anomalous data, a result or observation that deviates from what is normally expected. In experimental results, it normally refers to one repeated result that does not fit the pattern of all the others. Now you can see some graphs down below that shows outliers. This is that anomalous data. Now, why does it happen? Well, there could be errors in the measurement. Essentially, it could be, the scientist's fault. We didn't quite design the experiment correct. We're not measuring correctly, things like that. We didn't control the variables correctly, especially the control variables and the confounding variables. It could also be natural variation within variables being measured within the data. So there could be something going on we just don't understand. And that anomalous data is sometimes ignored when we calculate means and sometimes not. It kind of depends on the situation. Uncertainty is a quantitative measurement of the variability in data and categorized in two different ways. We can look at accuracy and precision. Accuracy is the correct value, how close the measurement is to the true or known value. Precision is the exact value, how close the measurements are to each other. So to example or demonstrate this, here's the bullseye test, right? Notice on the far left, you have all of the little X's on the bullseye in the middle, and they're all together. That's high accuracy and high precision. Now the second from the left, and you'll notice that all of the little X's are clustered together. That's high precision. They're all right there together, but it's low accuracy because it's nowhere near the bullseye. In the third down, you have high accuracy, low precision. They're all, circling the bullseye in that area, but they're not very precise. They're not right on the bullseye all collected together. And then, of course, the last one on the right, low accuracy and low precision. Now that we've talked about all this uncertainty, how do we decrease the uncertainty? Well, one thing you can do is choose the most appropriate equipment to collect the data that you need to collect. For example, using the smallest beaker possible to measure the volume of a liquid. Using the equipment properly, making sure, for example, you balance or calibrate the scale before you use it, zero balance, that kind of a thing. Controlling all the variables apart from the independent variable, making sure there's no variables doing something you don't know about. And of course, repetition. Repeat that experiment with similar results by other scientists. That's a really key thing. Now, when we talk about precision in measurements, the level of refinement in a measurement is shown by the number of significant figures given. And we should all be familiar with what significant figures are and how to work them. And there's just a diagram below to remind you of the different categories of leading zeros and trailing zeros and all that stuff. Now, if you're able to measure to the nearest 1 degree Celsius, then you can't record the results of 64.563 degrees Celsius, right? Because you can't go that many digits past the decimal. Now, you can use the same number of decimal places for every result. That's important. So if calculations were 43.34221 and 37, you would record them as 34.3 and 37.0. So you have to consider these various, norms when you deal with data. Now from accuracy, we're going to talk a little bit about integrity, right? Research methods must ensure the accuracy and integrity of the data being collected. The flaws in the methodology can lead to invalidation of that collected data, which means it's not really reliable. You also have to be careful about personal bias and making sure that that doesn't invalidate the research as well. No one experiment proves a hypothesis, only supports or refutes. And this is really important. We need to run experiments over and over and over and over again. Scientists must share their empirical data and methodologies and conclusions so others can read it and understand and try the same experiment themselves. That's peer review of empirical data, which is essential. Other scientists reading over your work to check it. And that protects the integrity of scientific advancement. It's really important, but it is a slow process. I feel like I would be negligent without mentioning the term theory. A theory is a broad natural explanation for a wide range of phenomena. It is strongly supported by many lines of empirical evidence. It may be modified or overturned if warranted by new evidence and perspectives. Essentially, it is the most accepted explanation based on extensive experimentation and multiple lines of empirical evidence. When we raise something from the level of a hypothesis to a theory, that means that hypothesis has been tested over and over again and nobody's been able to refute it. It has been sometimes tested hundreds and thousands of times by many different scientists over long periods of time. A theory is something that is incredibly sturdy within the scientific framework. Things like the atomic theory. We look at a theory of evolution. These are all examples of things that we have been testing and looking at for a long time. Now, that does not mean that the work is done. That does not mean that we still are learning about these theories and qualifying certain things and clarifying certain things. That is an ongoing process, and you can see that because scientists share this information all the time. They're back and forth and they're talking with each other. And that, of course, is the greatness of the scientific method. Really sharing that data, everybody going after that information and trying to run experiments and trying to understand the world around us. All right, that is it for us today. Those main ideas. Experimental design includes the formation of a testable hypothesis, the identification of variables, and the design of an experiment to collect appropriate data that is reliable. Scientists have the responsibility to act ethically and safely during any investigation. And of course, until next time, keep learning.